Good morning, good afternoon, and good evening to everyone joining from around the world. My name is Sabrina Welsh, and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. It's been an interesting week in the US, to say the least, and I haven't reported on the number of COVID cases in the last few meetings, so I thought it would be a good place to start this morning. I looked at the Johns Hopkins tracker this morning. The number of confirmed cases is almost at 48 million worldwide. It was about half that less than 2 months ago. And it's really sobering and scary to think about how the pandemic will progress in the next few months. There is reason to be hopeful, however, and today we're going to hear from one researcher leading the charge at the NIH in the development of a COVID vaccine. Just a note before we begin, the information presented today includes some pre-published data that is currently under peer review. At the request of the author, we ask that you not reproduce or disseminate the data presented. This session will be recorded and available on our website and social media for you to review. Today's speaker is Dr. Kizzmekia Corbett. Dr. Corbett is a research fellow and the scientific lead for the coronavirus vaccines and immunopathogenesis team at the National Institutes of Health, at the National Institute of Allergy and Infectious Diseases, at the Vaccine Research Center. She obtained her PhD in microbiology and immunology at University of North Carolina at Chapel Hill in 2014. A viral immunologist by training, Dr. Corbett uses her expertise to propel novel vaccine development for pandemic preparedness. Appointed to the VRC in 2014, her work focuses on developing novel coronavirus vaccines, including mRNA 1273, a leading candidate vaccine against the virus that causes COVID-19. The vaccine concept incorporated in the mRNA 1273 vaccine was designed by Dr. Corbett's team from Viral Sequence, and rapidly deployed to industry partner Moderna for an FDA-approved phase 1 clinical trial, which unprecedentedly began only 66 days from the Viral Sequence release. Following promising results in animal models and humans, mRNA 1273 is currently in a phase 3 clinical trial. Alongside mRNA 1273, Dr. Corbett's team boasts a portfolio which also includes universal coronavirus vaccine concepts and novel therapeutic antibodies. Additionally, Dr. Corbett spent several years working on a universal influenza vaccine, which is slated for a phase 1 clinical trial. In all, she has 15 years of expertise studying dengue virus, RSV, influenza, and coronaviruses. Along with her research activities, Dr. Corbett is an active member of the NIH Fellows Committee and an advocate of STEM education and vaccine awareness in the community. Combining her research goals with her knack for mentoring, Dr. Corbett aims to become an independent principal investigator. In today's lab meeting, Dr. Corbett is going to talk about SARS-CoV-2 mRNA vaccine development enabled by prototype pathogen preparedness. During the presentation, please send me your questions using the Q&A function in Zoom, and I'll ask our speaker a broad selection of your questions after she's finished her presentation. We'll have about 20-25 minutes for questions, and I hope you'll take the opportunity to discuss the data with us presented today. It is now my pleasure to introduce Dr. Kizzmekia Corbett from the National Institutes of Health. Dr. Corbett, good morning.
Good morning. Thanks for that introduction. And also the sobering reminder that we are still in a pandemic, and it is obviously not going anywhere without the help of a vaccine. And so I actually decided to today talk a little bit about how we got here by way of the vaccine development process, because I think it's really interesting to note, you know, how we got to this place. And I think that for the most part, many people are fairly staying somewhat abreast of the data that comes out of at least the preclinical and early phase clinical trial efforts. So I am Kizzmekia Corbett, and I am a senior research fellow at the Vaccine Research Center at the National Institutes of Health, which is a United States government entity focused on vaccine development for largely HIV and influenza, but also for vaccine development under this umbrella of pandemic preparedness. And so our coronavirus vaccine efforts stand under that kind of umbrella of vaccine development, and the work that we did for coronaviruses for years led to the point of which we were able to rapidly deploy a mRNA vaccine candidate in collaboration with Moderna Pharmaceuticals, and that is the work that I'll talk about today. Coronaviruses are a global health threat, largely because it's a large family of viruses that generally dwell in animal reservoirs. So coronaviruses, which are typically bat viruses, have the ability to transmit to humans, and in doing so, they cause widespread epidemics, and in the case of SARS-CoV-2, obviously a global pandemic. There are also coronaviruses that are endemic to humans, which cause more mild and flu-like symptoms seasonally around what we call flu season. But because of our knowledge of SARS-CoV-1, which circulated over a decade ago, causing 8,000 cases worldwide, and also MERS, which started circulating in 2013, causing over 2,000 cases worldwide, we obviously understood the imminence of the global health threat that coronavirus family caused. And this isn't just something that epidemiology has showed us time and time again, but something that laboratories that focus on viral evolution have pointed out in very clear manuscripts leading up into this point. Really, a large body of work coming from the Barrick Laboratory at the University of North Carolina suggested that SARS-like viruses were particularly poised for human emergence. And in our knowledge of that, we understood that there were three things that we needed from a vaccine solution for coronaviruses. The one thing, firstly, is that something be fast. In the case of a pandemic, we would want a vaccine technology that had the ability to be produced in vast quantities quickly. Secondly, reliable, a vaccine technology that could have been tested and upheld a level of manufacturability standard. And then universal. And I do have this italicized because I come from the universal flu field, and where the idea about universal vaccines is that one might be preemptive and provide protection against future outbreaks. And while we're not there yet for the coronavirus field, it is important to note that the way that we started to think about pandemic preparedness for coronaviruses is around a universal, considerably plug and play method for vaccine design. So without really belaboring the point, because Sabrina gave a very good introduction to what's happening worldwide with the coronavirus SARS-CoV-2 and with the global pandemic, it is of note that at this time, it would obviously take a vaccine to change the dynamics of the virus spreading across the globe. Of second note is that this SARS-CoV-2 sits in this beta coronavirus subfamily, and it is akin to not just SARS-1, but also to WIV1, which was deemed poised for human emergence, as I showed on my previous slide. And so in giving that, the type of approach that we take for vaccine development for pandemics, one of them is the prototype pathogen approach. And with this approach, the idea is that you study one virus in a particular viral family, and that would be your prototype virus, and you generalize knowledge around, around that particular prototype virus. And then you have the ability to apply that knowledge to multiple viruses within that virus family. And then the second prong of this prototype pathogen approach is that you would develop countermeasures at least through phase 1 clinical trials, which would increase the speed of development shall a pandemic occur. And so the SARS-CoV-2 vaccine development efforts that we've been taking on, and the globe actually, in fact, have been taking on, are part of a proof of concept around this prototype pathogen approach. And so with that, the spike protein that's on the surface of coronaviruses is a fairly well-conserved spike protein that's on the surface of all coronaviruses. And its purpose is to facilitate attachment of the virus to the host cell. And in doing so, the receptor binding domain of the spike protein binds to the human cell functional receptor. And then the fusion machinery, which is called the S2 machinery, facilitates fusion and downstream viral infection and replication. And so from a vaccine standpoint, targeting immune responses, particularly antibody responses that I'll talk about today, towards a spike protein was the goal. The problem is that for a large time, particularly before this pandemic, we had no idea about anything in regards to the spike protein other than that it was the target for vaccine development. And so for several years, prior to the SARS-CoV-2 pandemic, mostly beginning in 2013, we started to try to understand intricate details of how to elicit immune responses to the spike protein. And that work kind of came in several flavors. But mostly what happened is that we amassed this level of collaborative efforts with Moderna, which is the pharmaceutical company that we're working closely with for mRNA 1273, but also academic laboratories, and which all together kind of led us to the point of which we were able to rapidly develop a SARS-CoV-2 mRNA vaccine candidate. And so I want to walk us through this timeline in some fashion throughout this presentation, firstly starting with the extensive work that we did on MERS coronavirus and other coronavirus prior to SARS-CoV-2. And so the idea and understanding about spike proteins for coronaviruses, which is a fusion protein and akin to, for example, influenza HA, respiratory syncytial virus F, etc., is that when you can stabilize these fusion proteins in their pre-fusion conformation. So this is the conformation that sits on the surface of the virus prior to fusion and generally exposes more neutralization sensitive epitopes. And the proof of concept behind this idea or phenomenon really came about via the respiratory syncytial virus F and understanding around that protein, where you can see that within the pre-fusion conformation, these red surfaces or epitopes or antigenic sites on the surface of the spike of the fusion protein are exposed, but when in its post-fusion conformation, those red surfaces are occluded. So for vaccine response, if you want to target those more neutralization sensitive epitopes, you would want to expose, in this case, an animal, but people to the pre-fusion stabilized version of that protein. And so when I got to the Vaccine Research Center in 2014, there was no high resolution structure for coronavirus spike proteins. And so we set out to start to understand spike proteins from a cryo-electron microscopy standpoint with our collaborators at Scripps and also at Dartmouth. And we solved the first structure for human coronavirus HKU1 spike protein. And then there's this onslaught of literature with various groups around the world really understanding intricate details of coronavirus spike proteins, whether it be the glycans and epitope masking, the way that the spike protein moves from the pre-fusion to the post-fusion conformation. But lastly, we started to, as a field, begin to understand how the MERS and SARS, which were the most relevant from an epidemiology standpoint, spike proteins function. And in understanding that, we were able to describe from our laboratory what's considered to be a rationally designed pre-fusion MERS coronavirus spike antigen. And so the way that we went about that, I will get into some details, but it is of note that in rationally designing this pre-fusion stabilized spike antigen, really producing high quality protein really starts as the beginning for most everything as we think about these pandemic responses to not just by way of vaccines, but also by way of isolating antibodies for therapeutics, such as what was done in collaboration with Eli Lilly and our laboratory and also AbCellera. And then for use in diagnostics. It is also of note that the pre-fusion stabilized version of the SARS-CoV-2 protein, spike protein, is not just being used in Moderna's vaccine candidate, but in 5 of the 6 vaccine candidates that are currently furthest in human phase 3 clinical trial. So how do we get to that point? So we started with what is called structure-guided stabilization of pre-fusion MERS coronavirus spike. Having known the structure of several coronavirus spike proteins, and here I'm showing the HKU1 spike protein schema, and then really honing in onto the region around where the fusion peptide lays, which is in the S2 region, and coincidentally, in this S2 region of the spike protein is where much of the genetic sequences across the beta coronaviruses are similar. And so if we were able to look at how the spike protein for HKU1 is structured, we were able to see that there was this region in the S2 region where this hinge of the central helix and heptad repeat 1, which in the case of post-fusion would flip upon itself. We were able to put 2 proline mutations in that hinge region that functionally stabilized the MERS coronavirus spike protein in its pre-fusion conformation. And how do we know that? Well, firstly, because in cell culture, the 2 proline version of the spike protein expressed to 50-fold greater than the wild type version of the spike protein. But then by electron microscopy, if you express the 2 proline version of the spike protein, or S2P, you get this mushroom-shaped homogeneous trimer spike protein, as opposed to with expression of the S wild type, where the spike protein spontaneously flips in its hypothetical post-fusion conformation, where the shape of the protein is more elongated, as you might expect, as this spring flips upon on itself. And so because of the genetic similarities across the beta coronaviruses, particularly in that S2 region, putting the 2 proline mutations into the backbone of HKU1 and SARS, along with MERS, stabilized it in its pre-fusion conformation. And the same is true with several other coronavirus spike proteins as well. And I will just point out that what was interesting then is that the same was true for the WIV1 spike protein, which was deemed to be the potentially emerging coronavirus. And so because you have this, what is considered to be plug and play, where the 2 proline mutations can be slotted, so to speak, into the backbone of spike proteins across the coronavirus family, or at least beta coronavirus family, the next step is to figure out how you might want to deliver that spike protein in the case of a pandemic. There are obvious downsides to particularly with speed, as you think about subunit protein vaccination, et cetera. So we took advantage of a collaboration that we had with Moderna for developing other types of vaccines. And so with their platform, they can insert a new therapeutic protein. In this case, it would be the 2 proline versions of spikes for coronaviruses. Place it into mRNA sequence, and then put that mRNA sequence into a lipid nanoparticle formulation. And then by using their mRNA lipid nanoparticle and delivering it into the muscle, you basically tell the body to make its own spike protein, bypassing the step to make GMP grade spike proteins for delivery. And so that increases the speed of the technology. And because Moderna had tested several vaccine candidates through phase 1, in an early stage of phase 2, also the manufacturability standard was there. And because of the ability to kind of drop any protein into this system, the italicized universal vaccine technology still remained. And so we use the MERS S2P as the prototype pathogen approach with the mRNA as proof of concept that you might deliver a S2P antigen via the mRNA platform. So in collaboration with Ralph Barrick's laboratory at the University of North Carolina, where they have a lethal challenge model for MERS coronavirus, we were able to vaccinate those animals and then challenge with MERS coronavirus. And you can see that at very modest doses of MERS S2P mRNA, even down to the 0.1 microgram dose, you completely protect these mice from weight loss following a lethal challenge. And even at what is considered to be a subprotective dose of 0.01 microgram, the mice still recover. And we didn't just see that with weight loss, but also with lung viral titers and lung hemorrhage, which is a way to score the amount of lung disease that these animals have. And so what this showed us is that MERS S2P mRNA elicited dose-dependent protection against a lethal MERS challenge and without evidence of any enhanced illness. And so it seemed that we had a system by which we could deliver both a precision-designed antigen via a platform that could be rapidly developed. And so when the sequences of at the time, 2019 N coronavirus is what it was called, came out on January the 10th of this year, we were able to decide with Moderna on a sequence for the mRNA 1273 sequence just 3 days later. And we were able to move very quickly to really understand the immunogenicity of mRNA 1273 in mice. And while I will not have the time to show you the data from those mouse studies today, they are published in Nature in this manuscript here, which took a large collaborative effort specifically from with UNC's Ralph Barrett's laboratory. What I will show you though is some of the data that we got from our studies in non-human primates, where we vaccinated rhesus macaques with mRNA 1273 at week 0, and then following that with a boost at week 4 to challenge the animals with close to 10 to the 6 PFU of SARS-CoV-2 4 weeks following the challenge, and then looking at not just the challenge outcomes, but assessing the antibody and T cell responses as well. So the doses that we chose to use in the NHP model were the 100 microgram dose of mRNA 1273, which can be considered a so-called clinically relevant dose because that is the dose that has moved forward into the phase 3 clinical trial, and then tenfold less with a 10 microgram dose of mRNA 1273. And here I show the temporal antibody responses both for spike-specific binding IgG and then also for pseudovirus neutralizing antibody titers as well. So you can see that following the first dose of mRNA 1273, you get this boost in spike-specific binding IgG titers that remain steady through the second dose. And then following the second dose, you get an increase of mRNA, of antibody titers following mRNA 1273 vaccination. So you get up to 10 to the 5th spike-specific binding IgG titers. And those spike antibodies, as we showed in a separate figure, correlate with the neutralizing antibody titers that we see. So the higher neutralizing binding titers means the higher the neutralizing antibody titers, where you get this modest neutralizing antibody response up to 10 to the 3, actually following a 100 microgram dose of mRNA 1273. That is increased upon following a second dose of mRNA 1273, and at least remained steady through to the point of which we challenged the animals at 4 weeks following the second vaccination. So it's interesting to note that we did see binding IgG titers and pseudovirus neutralizing antibody titers 4 weeks following the second vaccination. One thing that we wanted to show, particularly with these data, is that not that we were just eliciting neutralizing antibody responses, but how does that, the types of antibody responses that we are eliciting with mRNA 1273 compare to that of human convalescent sera? So sera collected from people who had recovered from COVID-19 disease. So in the purple, with the purple dots here on the far right of each of these graphs are those serum, and as you can see, with 100 micrograms of mRNA 1273, you elicit immune responses that are in the upper quartile of what you see in those human convalescent sera. And while even the 10 microgram dose of mRNA 1273 in monkeys is more modest, you still have fairly high antibody responses, and particularly as you start to look at more functionally relevant antibody responses. So these assays in panels E and F are an ACE2 binding inhibition assay, where you're essentially asking the question, how much of the antibody is blocking the ACE receptor binding domain from binding to the ACE2 receptor in vitro. And we are eliciting a great amount of ACE2 binding inhibition in both the 100 microgram and 10 microgram groups of mRNA 1273. As well as with a live virus neutralization assay. In this case, we chose a nanoluke neutralization assay. And with in this case, we're also beating out the ability of the antibodies from convalescent sera to neutralize live virus. And so that was really good to see, because we could not only show responses at 100 micrograms, but at a more modest dose in the NHP model. And it became then clear to us that we were most likely eliciting the types of T cell responses that we wanted to elicit, particularly as you think about vaccine-induced enhancement for other respiratory viruses. So T cell responses have been shown to drive vaccine-induced enhancement if they are not optimal. So in the cases of coronaviruses, particularly in some of these earlier preclinical studies and in the phase 1 clinical trial, you really wanted to show that you were eliciting a Th1 skewed T cell response. So here in this figure, what we show from those monkeys is exactly that is what's happening. So the red dots in each of these figures, or the blue dots, are the responders of each of the T cell subsets in each of the NHP groups, whether it be 100 micrograms for red or 10 micrograms for blue. And you can see that 7 out of 7 of the monkeys that got mRNA 1273 at 100 micrograms respond and have a Th1 CD4 T cell response, as opposed to only 2 of those that have a Th2 T cell response. And obviously, the Th2 T cell response is minuscule as compared to the Th1 response, which showed us that we were eliciting the type of T cell response that we'd hoped for, as well as when you look at TFH cells, that we are eliciting robust TFH responses in these animals, which kind of correlates with what we see in so many ways with the antibody responses being so great. So it was not just that we were moving in a timeline towards understanding the immunogenicity in these animal models, but at the same time, we had initiated with Moderna cGMP production of the phase 1 clinical trial material. And so that started immediately after we decided on mRNA 1273 sequences. And then Moderna was able to ship that drug product out to for clinical trial only a little over a month following initiation of production, which really shows just how robust these types of gene-based platforms can be as you think about rapid vaccine development. And then the phase 1 clinical trial started only 66 days after the SARS-CoV-2 sequences were released. And so I will show just a snippet of the antibody data from the phase 1 clinical trials that began. So there were 2 phase 1 clinical trials that began, one of which that was in Seattle and it was led by Lisa Jackson. And then the second of which was at Emory, which was led by Dr. Anderson. Combining those 2 phase 1 trials together in this table, we dose people with either 25, 100, or 250 micrograms and had a young adult cohort from 18 to 55. And then with 25 and 100 micrograms of mRNA 1273, older people were given the vaccine as well. And so the design of these studies was really to show 2 things. How does the response differ across the various doses in humans? And then to also inform the types of doses that would move forward into the phase 2 and phase 3 clinical trials. And then also, and importantly, given how severe disease from COVID-19 is in elderly people, how does this vaccine fare in more aged populations? And so we, similar to the NHPs, gave doses at week 0 and week 4 with these various doses, and are following these people for up to a year. And while I don't have any of the durability data today to show you, because it is as yet unpublished, although hopefully coming out very soon, I do have some preliminary data from these for antibody responses in these people following their second dose of vaccine. So firstly, with the younger adults, again, we assess the S-specific binding antibody responses. There are a couple of things that I want you all to take away from this graph. The first of which is that for binding antibody responses, while there is a dose response, but you are eliciting very robust antibody responses even at the 25 and 100 microgram doses of mRNA 1273. The second of which is that it is very clear that there is this prime boost response that is happening. So after the first dose of mRNA 1273, you get about 10 to the 4 endpoint titer for S-specific binding antibodies. That is increased by a log after the second dose of the vaccine. And these antibody responses are remaining steady at least through day 57 of the phase 1 clinical trial. The other thing that is of importance to note from these graphs is that, again, comparing to a panel of convalescent sera that was collected from people that have recovered from COVID-19 disease, we are eliciting very high immune responses in the upper quartile of what you see in humans that have recovered from COVID-19. And with the pseudovirus neutralizing antibody responses, the same story remains true. You get this modest response, and even maybe even some of the people do not have responses after the first dose of mRNA 1273. But after the second dose of mRNA 1273, 100% of the participants in this small 45-person phase 1 clinical trial respond with robust neutralizing antibody responses to SARS-CoV-2 spike protein, but also that they are in the upper quartile of what you see with human convalescent sera. But what does that mean for older adults? And I'm happy to report, and this paper came out, I think probably about a month ago now, that the amount of neutralizing antibody responses that we saw with the 100 microgram group, which is again the dose that is moving forward and is in phase 3 clinical trial, in the people that are between the ages of 18 to 55, that people that are over the age of 56 have similar neutralizing antibody titers following the second dose of mRNA 1273. So all in all, we are eliciting high levels of neutralizing antibody responses in not just younger adults, but also in older adults. So in conclusion here, there are several coronavirus spike chimers that can be stabilized in their pre-fusion conformation by inclusion of the 2P mutations. That includes the SARS-CoV-2 spike protein, although we really showed a lot of immunogenicity data moving into this point with a prototype pathogen, the MERS coronavirus spike. And the inclusion of those 2 proline mutations has been shown to be highly immunogenic and protective in animal models for mRNA 1273, but also in preclinical studies for several of the other vaccine candidates as well. And mRNA 1273 elicits robust immune responses in humans, particularly as we think about antibody responses. And I didn't show any T cell responses, but we are also getting CD4 T cell responses in humans as well. mRNA 1273 is currently in a phase 3 efficacy trial, where as of, I think, about 2 weeks ago, the phase 3 clinical trial was completely enrolled with about 30,000, a little over 30,000 people. And so we are on the way to hopefully get an efficacy signal from the phase 3 clinical trial very soon. And without further ado, I want to thank this small team first in the middle, which is the team that we started with at the Vaccine Research Center that has been studying coronaviruses over the last, even before me, 10 years, but since me for 6 years, led by Dr. Barney Graham and also by Dr. John Mascola. And since the pandemic has occurred, VRC-wide, there have been all hands on deck that has helped in so many aspects, not just with mRNA 1273, but with other vaccine candidates as well. The animal studies, particularly the NHP studies, have had so much help from Bob Cedars' laboratory and also from Mario Roeder's laboratory. We have been collaborating with several labs across the United States since our ideas around prototype pathogen design with MERS, largely with University of Texas at Austin, Jason McClellan's laboratory, and Andrew Ward's laboratory at the Scripps Research Institute. And then for some of our next generation candidates with Neil King's laboratory, we work very heavily with Ralph Barrick's laboratory with using their mouse challenge models to really understand efficacy of our vaccines in full. And for the neutralization assays, while I didn't show any of the data here really today from many of the neutralization assays, but a lot of the work that's been put into the phase 3 neutralization assays comes from David Montefiori's laboratory at Duke University, and the ACE2 cells that we got from David Ho were instrumental in rapid development of our pseudovirus neutralization assay. And without further ado, I will now take questions.
Dr. Corbett, thank you so much for that wonderful presentation. It's incredible. I mean, it's an enormous amount of work and ingenuity and creativity that led us here. And it's great to see all the partners that came together to collaborate. I've seen a lot of the Human Vaccines Project research teams working together. It's really inspiring. So thank you for sharing that with us.
Thank you. Yeah, I mean, I think, you know, one of the things that I remember most when I do have a chance to actually play out how all of this has worked on my downtime, one of the things that I remember most is almost coincidentally, the viral sequences came out on January the 10th. And then that week was this week that we had collaborated meetings with Moderna and UNC and Vanderbilt, et cetera, that were just already on our calendar because we've been working with them for so long. And it was almost kind of like the universe was was really telling us that, you know, you're ready to go, and so go.
Well, and clearly that model of collaboration really pays off and really shows us what we're capable of when people work together and think creatively like that together. It's fantastic to see. We're getting tons of questions. Thank you so much to our audience for being so engaged. I'm going to start on the antibody side of things today. What's the ratio of the net antibody to total antibody that you saw?
I don't think I understand that question, but do they mean the neutralization antibody?
Likely. It was, let me see.
Let me see if I can read it. I don't.
That was from Jill Gilmore, one of our one of our colleagues from IAVI, who has been in this field for a long time and keeping an eye on this data. Jill, maybe if you want to clarify your question. Yeah, the ratio of net to total antibody.
So I don't know if I understand that question, but hopefully I can answer it in the way that I think. And Jill, if that's my answer does not answer you, then
oh, so she's clarifying the total antibody. How much is neutralizing antibody?
Okay, that's what I thought she was saying.
Thanks, Jill.
Yeah. I didn't show the graph, but at least so we mostly for the most part have been characterizing things in full with our pseudovirus neutralizing assay. And so with that, what we showed in the mouse paper is that once you get to a threshold of about 10 to the 4, 10 to the 5, spike-specific binding antibody titer is when you start to see these really robust neutralizing antibody titers, and they correlate with each other. And so that's something that we not only saw in the mouse studies, but something that we saw in the human studies and NHP studies as well. And so one of the things that is, I think, somewhat in the back of people's mind when they ask that question is that because we are giving a full-length spike protein, right? And for the most part, the most neutralizing of those antibodies directed towards a spike protein, or the most potently neutralizing antibodies, are towards the receptor binding domain. And so do we see non-neutralizing antibody responses in this kind of polyclonal amount of antibodies that we are eliciting? And we do see antibody responses that are targeted outside the receptor binding domain. We haven't really dissected out how much of those are neutralizing versus not. But with the level of neutralizing antibodies that we are eliciting, not just with mRNA 1273, but with the other full-length spike protein vaccine candidates as well, and with the amount of ACE2 binding inhibition, we don't really think that the non-neutralizing antibodies will be a problem. And if anything, they likely work together via other mechanisms that haven't been worked out towards protection.
Mm-hmm. And this is sort of a related question. There appears to be a range of cross-reactive binding antibodies to the S prior to vaccination, to the S protein. Are there differences in responses post-vaccination if these individuals were stratified by either the absence or presence of the cross-reactive binding responses prior to vaccination? Sort of a complicated question.
It's not that complicated. And
okay.
someone is like writing my aims in my head for me, right? So assuming that I'm assuming that they've, yeah, I mean, like, everyone's seen the literature, but yeah, so that's actually one of our questions is if the pre-existing antibody signature of what people have, whether it be for the other, particularly endemic coronaviruses, how that might infer the type of vaccine responses that one gets afterwards. It's actually even more of a question now because of seeing the younger cohort versus the older cohort, right? So people who are in their 70s have obviously been exposed to more endemic coronaviruses and might have a different type of antibody signature or B cell signature prior to coming into the trials. And so that's a very good question, but we don't have an answer yet.
Yeah, that immunologic imprint that older people might have prior, to receiving this vaccination could be significant. I wonder, are you doing imprinting type studies or samples for that type of work available?
Yeah, I'm not doing it, but the VRC has plans to do that, mostly with the guru who anything any do it.
Mm-hmm.
Yeah.
Super. Okay, more questions on serum binding and neutralizing antibodies. mRNA 1273 induced high levels of Th1, but low levels of Th2 cell responses. Do you see high levels of serum binding and neutralizing antibodies, and then how would you explain that?
Do I see high levels of serum binding and neutralizing antibodies? Yes, I
think that's, yeah, that graph that you previously showed.
Yeah, that was the data that we showed. Maybe there's a part of the question that I'm not understanding, and if so, then I'm sorry. Okay. But yeah, so we see very high levels of serum binding and neutralizing antibody titers, and we haven't necessarily correlated it with the type of T cell response or anything like that just yet.
Mm-hmm. Okay, great. Let's see, I'm getting a couple of technical questions. These may be easier to answer offline, and I'll share your questions with Dr. Corbett afterward. Let's see. Okay, so we're getting a lot of comments. Your work is so impressive. Are you trying to further improve the responses of 1273, for example, by using Hexapro instead of S2P? And have you mapped the serum responses in more detail to the spike? People are very curious about that.
I know. So this is the thing. I like, if people are on this call and ever saw me present prior to the pandemic, you know that I would like come to meetings with data that like the team generated overnight before. Unfortunately, I'm not in a position to do that
Sure.
At this time. And so, there are so many questions that we want to answer. It's obviously very important to map the antibody responses. It is obviously also really important to understand whether there might be any utility to next generation vaccine design, whether it be Hexapro or other. And so those types of things are in the works.
Definitely. Yeah, and there's a lot of data to come. I'm sure there's tons of experiments. If you had all the time in the world, they would design right now, but they're coming, guys. Okay, question about the half-life now. Is the half-life of the mRNA in the receiving muscle cells determined?
Long story short, no. Yes, no.
Okay. Let's see. There are a couple questions about the dosing of the mRNA product. Does mRNA continue to produce the antigen in the cells? Is there a need for multiple doses, or would one dose? It looked like the response after the boost was tremendous. So that seems pretty self-explanatory, but.
Yeah, so the response after the boost is, so if you remember, particularly in the humans, actually, let me just back up. I'm gonna talk about the animal data first and kind of work my way back to the humans. So with in the mouse studies, what we saw is that you can elicit neutralizing antibody responses, with 10 micrograms or 1 microgram in a mouse after 1 dose, and that those antibody responses or the immune response that you elicit is protective. So after 1 dose in a mouse, up to 7 weeks after 1 dose, you can protect against, challenge with a mouse adapted strain of the virus. Mm-hmm. In the humans, that looks different, right? Because as you saw in that graph, after 1 dose, there were some people who didn't get a neutralizing antibody response at all. Whereas after the second dose, 100% of the people do have a neutralizing antibody response. So understanding what the utility of 1 dose might be from the mRNA 1273 vaccine candidate is something that we have to figure out, and how those data from animals might correlate with humans, and whether or not even in the case that there aren't any new quote unquote neutralizing antibodies, at least as you can show with the types of new assays that we do, whether those types of people would be protected, might come out of the more large scale phase 3s where, people might drop out of the trial before their second dose, or by chance get infected before their second dose. And so hopefully some of those data might come out.
Great. And just related to future studies, I don't think the phase 1 studies, the mucosal immunity is being assessed. Would a mucosal study or maybe a sub-study be considered for future clinical trials?
No. We're not planning
the question from the person was, is there any mucosal immunity response assessed? But I think that's not being done currently.
Yeah, we're not looking at mucosal immunity in this case because we don't necessarily think that we need to go there in the case of SARS-CoV-2. And the way that a true bona fide mucosal immunologist would think about mucosal immunity for sure. And it would likely take, delivery of the vaccine in a different way, whether it be, into the nose, et cetera. But we're not planning to look at that at this time.
Okay. And just related, you may have, this may be part of the data that you didn't show. Are there any plans to test Th1 responses by ELISpot or ICS?
We did that. So the data that you saw today were ICS data. I'm sorry I didn't make that the assay that we use more clear. We don't use ELISpot, we use ICS. And so those were spike-specific Th1 responses that you saw both from the NHPs and, I didn't show the humans, but the same assay is used for humans as well.
Great, okay, thanks for clarifying, that's helpful. We have a couple of questions about the durability data. I don't think we have yet the immunized human data available for durability. Is there longer term durability information about the immunized macaques? Or from any of the animal studies, I suppose.
Wait, so I want to back to the ICS question in that
yes.
Only to say that the cytokine profile for NHPs and the mice are all published in those papers. And then going back to the question that you asked about durability in the NHPs, well, we have started durability studies. I think I can say that out loud. I'm pretty sure it's in the discussion.
Great. Breaking news. I'm glad meeting.
I know it's not breaking because it's obvious. It's the logical next step, right? You will obviously get some durability data out of the phase 1 because we're following people for a year, but in a more controlled challenge way, we definitely are looking at durability in the NHP studies. So those data are not out yet.
Great. Okay, so looking at sort of a different angle about the research now, could you comment on R&D funding? There's a lot of talk about pandemic preparedness and maybe we should be restructuring to increase the biomedical pandemic preparedness response in our country. If research on a prototype pathogen can create benefits for work on other pathogens, would this suggest that pathogen-specific funding approaches maybe are inadequate, or there should be cross-disease or cross-fields funding?
To sway the response away from what can consider to be advisory. From my position, I'm obviously not in a position to advise where funding should go. But Dr. Graham and I did write the commentary on prototype pathogen and pandemic preparedness And so the idea is that, yes, it would be a bulk of funding that would need to go into these 24 viral families that have the potential to emerge as pandemic threats.
Mhm.
What the amount of that funding looks like, what family gets more versus the other, not going to comment there. But that, yes, the reason why this type of work has proven to be fruitful is because of essentially because of the resources that were poured into it prior to the pandemic. And also, I guess one could say that the reason why the getting into the phase 1 clinical trial wasn't even faster, although 66 days is admirable.
It's remarkable.
It's because that there was no MERS spike, for example, phase 1 clinical trial. So one of the prongs of the prototype pathogen approach is to move things through phase 1 clinical trial even, which takes an even larger investment. Mm-hmm. And so in an ideal world, if I had control, then yes, funding towards the various viral families, having some level of modeling around which prototype pathogen from each of those families would need to be studied most intensely and blah, blah, blah. I think that totally would be the way to go.
Great. Yeah, you know, it's been interesting to see the different collaborations. And I mean, the HIV and influenza fields have clearly kind of fed into this. You know, a lot of the infrastructure and the testing and the assay development was there, which I think helped, you know, the labs spur along these types of developments. It's amazing.
Absolutely. I mean, I think, yeah, I mean, people have really leveraged individual laboratories and also collaborative efforts have really been leveraged off of previous work, whether it be flu, HIV, et cetera. I mean, minute details, even like our pseudovirus assay, how we really got it up and running. We obviously had a MERS pseudovirus neutralization assay, but that really all came to fruition because of our influenza pseudovirus neutralization assay.
Sure.
So there really is this kind of intricate network of scientists that are already doing pandemic preparedness without even calling it that, to be fair.
Yeah, sure. Could you comment a bit more and just tell us your experience about the NIH and Moderna collaboration? One of our participants commented, it's really interesting to hear about the key role of public research institutions, since we often think about vaccine product development as more of like a private sector endeavor. So it's great to see these types of private-public partnerships. Just curious for your feedback on the collaboration, what it's been like for you.
I mean, it's been great. So one of the things that is really, I think, unique about the Vaccine Research Center, and sorry that I was looking because I thought that I actually had another PowerPoint slide that introduces this actually, but I didn't. One of the things that's fairly unique about the VRC is that the entire goal of the center is really to utilize what we understand from basic research, starting with even protein structures on a computer screen, to move those into phase 1 clinical trial as vaccine candidates. And we even have our own clinic to do that. And then the actual idea is to then allow industry to take over from there. The Moderna collaboration is really just kind of these years long of working together to answer fundamental questions about how to express the spike protein for coronaviruses. So things that I even didn't show here. I mean, we were working together to understand whether to make the protein secreted in its secreted version or whether to have it transmembrane anchored. What's the utility of S2P versus wild type? All these things in the context of their platform. And so the collaboration really kind of materialized in a very different way than what you would normally see. But you know, companies have the ability to develop things out into further trajectory than most academic and government institutions do. So it is kind of what I consider to be a beautiful marriage, so to speak, I think.
Absolutely. Yeah. Well, and you can see how much that impacts the speed at which things can move forward when you really have that partnership in place. It's great to see. Okay, you mentioned earlier T cell responses. I'm not sure if that data is available, but are CD8 T cells induced, and is there expected potential T cell responses?
So the T cell responses that I showed are the ones that are being induced.
Okay, sorry, we've missed that before.
At least given the methods that we use, which is, yeah. So I think we don't see any reputable CD8 T cell responses in the case of the NHP or the human. We did see CD8 responses in the mice that we looked at, but with note that with a varied assay and time points, et cetera, blah blah blah, that Pfizer's mRNA candidate, which is very similar to the Moderna vaccine candidate, they do see and report CD8 responses in their phase 1 human trial.
Okay, great. And could you comment on viral escape? There was a question, was there viral escape in the NHP cohort? And I think the second part of that question we answered. So yeah, if you could just comment on the viral escape element.
Yeah, so we did not sequence any viruses that might have escaped from the NHP cohort. So in the case here, we didn't see really robust viral replication after day 2 following challenge, particularly in the lung, in either the 100 microgram or the 10 microgram group. And so there wasn't much breakthrough.
Yeah.
And so we, would have to do more intricate studies where we plan for breakthrough and then sequence those viruses to look at what's escaping versus what is not.
Great. And just a question now on the observed adverse reactions in the volunteers. Can you comment on that? I'm not sure if that data is if you want to talk about that today, sort of more the clinical side, but are there side effects observed in the volunteers? And if you could talk about that profile a little bit.
Yeah, so I can talk about it briefly. So the phase 1 clinical trial, yeah, so there are, fever, et cetera, that has been observed, particularly after the second dose of mRNA 1273. And so this is standard reactogenicity that people observe even after, for example, an influenza vaccine. Mm-hmm. So the standard kinds of side effects that one might see, and those types of side effects were greater once you up the dose to a 250 microgram dose in the phase 1. Which is why the second phase 1 only has the 100 microgram dose, and while the 100 microgram dose is moved forward into the phase 2s and 3s. And obviously you're eliciting as robust antibody responses. And so 250 is probably too much anyway. And we're starting to think even 100 might be overkill, but at least,
yeah, sure.
Yeah. But yeah, you get standard types of side effects as we saw in the phase 1 clinical trial, and that has been reported, not just for our vaccine candidate, but like Pfizer's, for example, as well.
Mm-hmm. Great. Okay, we just have a couple more minutes. I'm gonna ask a few more questions that are a little more brief. Let's see. Okay, pretty straightforward. Do you know if the mRNA 1273 includes the D614G mutation? Meaning, is there a D or a G at position 614?
I love that.
Very specific.
I love that you're taking the questions and like rewording them. It's actually like very, I don't see people do SciComm well, so good job. But yeah, so yes, I do know, and it's a D at the 614 position, and we've tested serum from mice. That's published in the supplement of the Nature paper. Humans is published in, I think, the second preliminary report, et cetera, for viruses that have the G mutation, and we're eliciting actually better neutralizing antibody responses to the G virus.
Wow. Great. Well, that's encouraging. Any final thoughts? I just wanted to get your impressions. If you've have an upcoming data set that we haven't heard about yet that you want to maybe comment on, or you want to comment on next steps, or just what you're hoping to see in the next
you're a
few
great
months?
moderator, but you're also a reporter, apparently. This is
just trying to get the scoops. You know, we're all, everyone is so excited about the data. I feel obligated to ask.
It's fine. No, this is what I get a lot, and I have nothing to say other than that where all of the questions that people generally ask, I mean, you know, the things that you would want to know about durability and escape mutations and pre-existing immunity and how that fares with vaccine responses, all of these things are going to be in the works, and the plan is to publish things as soon as we know them. And so we are working to make sure that we answer people's inquiries around mRNA 1273 in particular. And I know these studies are not just coming out from like our laboratory, but also like from with other vaccine candidates from other laboratories as well. Unfortunately, I can't say what's in the pipeline and what's been submitted versus what's not, but
sure. Well, thank you so much for answering all the questions today. I think discussions like this are so valuable, especially when we can connect you directly with people who have questions. We tried to recreate this lab meeting type of environment. So thank you so much, Dr. Corbett, for your time. I really appreciate it. And thanks to our audience members for asking your questions today. We appreciate you being so engaged in the conversation.
Thank you. Bye.
Thanks. I invite everyone to join us 2 weeks from today on the 19th of November. We're going to have Dr. Eleanor Fish from the University of Toronto, who's going to speak with us about global virus outbreaks, interferons versus as first responders. So stay tuned, we'll be back in 2 weeks. And if you're interested in more information on COVID-19, please sign up for the HVP COVID report. In this newsletter, we present the latest data and scientific articles from around the globe. So if you are interested in more information, please check out the COVID report. And finally, you can find the full session of this webinar recorded on our website and on LinkedIn and all of our social media handles. So with that, I'll say stay safe. Thank you for joining. And we hope that we'll see you in 2 weeks on the next Global COVID Lab meeting. Thanks so much, everybody.